Have you ever wondered what actually happens after you eat something sweet? We're about to go on an incredible microscopic journey to find out. We're going to follow a single sugar molecule from your food all the way into your cells. And trust me, it's a journey filled with some amazing biological puzzles and some very, very important gatekeepers. Let's get into it.
So, picture this. You just polished off a delicious cookie. The sugar is now in your gut. It seems pretty straightforward from here, right? It just gets absorbed and gives you energy. But it's actually not simple at all. In fact, there's a huge problem. A massive biological wall that every single one of those sugar molecules has to figure out how to cross. And this is the big problem. Think about oil and water. They just don't mix, right? Well, that's exactly what's happening here. The cells that line your intestine have this outer layer, this membrane that's made of fat, so it's hydrophobic. It fears water. But sugar molecules, they're the opposite. They're hydrophilic. They love water. So a sugar molecule can't just stroll through that cell membrane. No way. It needs a special invitation, a secret password to get inside.
So how in the world does the sugar get in? Well, the answer lies with a team of highly specialized proteins. Think of them as the bouncers at an exclusive club, the gatekeepers of your cells. They're embedded right in the cell membrane, and it's their job to decide which sugars get in, when they get in, and exactly how they get in. It's time to meet the crew.
First up, let's meet the main workhorse of the team, SGLT1. This guy is the primary escort for glucose and its close relative galactose. Now, SGLT1 is what we call an active transporter, which means it has to spend energy to do its job. It has this really clever system where it harnesses the flow of sodium into the cell to literally drag sugar molecules in right alongside it. It's an incredibly powerful mechanism.
So, what makes SGLT1 so good at its job? It's all about its affinity. It has a very high affinity for glucose, which basically means it's super grabby. Even if there's just a tiny amount of sugar hanging around, SGLT1 will snatch it up to make sure your body doesn't waste a single bit of energy. And it's positioned right at the front door of your intestinal cells, working nonstop.
Okay, our next gatekeeper is totally different. This is GLUT5, and it is the fructose specialist. If glucose and galactose are using that main entrance with SGLT1, fructose gets its own private door. GLUT5 is exclusively dedicated to one job and one job only, getting fructose into the cell.
And unlike our high-energy friend SGLT1, GLUT5 is a bit more laid back. It uses a process called facilitated diffusion. It doesn't need any energy or any sodium to work. It simply opens up a channel, a specific doorway, that allows fructose to flow naturally from where it's crowded in your gut to where it's less crowded inside the cell. Simple and efficient.
All right, so SGLT1 and GLUT5 have done their job. The sugar is now inside the intestinal cell. But that's only halfway there. How does it get out the back door and into your bloodstream to be used for energy? Well, that's the job of our third and final gatekeeper, GLUT2. This is the versatile, all-purpose exit door. It can handle all three of the sugars we've talked about: glucose, galactose, and fructose.
Now, let's check out GLUT2's profile. You can see it usually hangs out at the exit, but look at that special skill because this is where it gets really interesting. During what you might call a sugar rush, when a ton of sugar hits your gut all at once, GLUT2 has this secret superpower. It can actually pack up and move to the front door, opening up a second high-speed superhighway for sugar to pour into the cell. This little trick is absolutely crucial, and we're going to come back to it.
So how do these gatekeepers' personalities compare? We can look at this value called Km, which is just a scientific way of measuring how grabby they are. And the key thing to know is a lower number means it's more grabby. So look at SGLT1 with a tiny Km of 0.5. It's the picky one. It'll grab glucose even if it's the last one at the party. Now look at GLUT2. Its Km is way up at 17. It has a low affinity. It's kind of lazy. It really only starts working when the party is packed and sugar is everywhere.
Okay, so we've met the team, we know their individual skills, but the real magic is in how they work together. So let's watch them in action. We'll compare what happens in your body after two very different meals and see just how brilliantly this whole system adapts.
And this is where the whole story just clicks into place. Imagine you eat a low-sugar meal, like a big salad. Your SGLT1 transporters get to work, diligently pulling in every little bit of glucose they can find. It's an efficient, careful operation. But now, imagine you chug a high-sugar soda. The system completely shifts gears. SGLT1 gets totally overwhelmed. It's working as fast as it can. And that is the signal for GLUT2 to race from the exit to the entrance, throwing open the floodgates to handle that massive sugar rush. It's an amazing response.
The key here is that this isn't just a random event. It's a beautifully coordinated chain reaction. When SGLT1 starts working at its absolute maximum capacity, all that frantic activity sends out a signal. It's basically like the main gatekeeper is screaming for backup. And in response to that signal, the cell physically moves its GLUT2 transporters to the front line. The result? The cell's ability to absorb sugar just massively and very rapidly increases, right when it needs it the most. Now, this system is so elegant and so efficient, but, it's also incredibly delicate. So what happens when one of these essential gatekeeper proteins just doesn't work? This isn't just a theoretical question. This has very real and sometimes very serious consequences for people's health.
To understand this, let's take a look at a specific rare genetic disease. It's called glucose-galactose malabsorption, or GGM for short. And it gives us this perfect, although really unfortunate window into what happens when that key gatekeeper, SGLT1, is broken from birth.
So in someone with JGM, there's a tiny defect in the gene that's supposed to build the SGLT1 protein. Their primary escort is just gone. And the result is that glucose and galactose, say from milk, get completely stuck in the intestine. They can't get in. All that unabsorbed sugar draws huge amounts of water into the gut, which for a newborn causes severe, truly life-threatening diarrhea. It really drives home just how absolutely critical SGLT1 is for our survival.
Understanding this whole intricate system of gates and doors isn't just a cool science lesson. It actually opens up some incredible possibilities for medicine. Because if we know exactly how the gates work, well, then maybe we can learn how to control them.
And guess what? We are already doing it. For instance, we learned that in type 2 diabetes, the body can actually start making more of these transporters, absorbing sugar even more aggressively than usual. Common drugs like metformin actually work, in part, by messing with the activity of SGLT1 and GLUT2. And huge interventions like bariatric surgery can physically change the gut in a way that leads to fewer SGLT1 transporters, completely changing how the body handles sugar. And all of this leads to one last, really compelling question for the future. Right now, we have these powerful but kind of blunt tools, drugs, surgery, to hack this system. But just imagine, could we one day use specific foods or even compounds in foods to gently tell our gatekeepers how to behave? Could we fine-tune our own sugar absorption every day, just with what we eat? The science is really just getting started, but the answer could completely change everything.